microphone 1 2. Welcome back to our class. Today we are going to be looking at some of the things which might come in the end of term 1 exam. So I'm going to refer you to our distinction material book and then just show you that you cannot go to the paper without doing this, without doing this, without doing this. I only have 20 minutes to smash this so that by the time you leave this live, at least you'll be having some questions which you might answer. I'm not going to be writing, I'm just going to be talking, and then make sure that you note it down what is important for you. And if you have not done it before, or you have not done it, try to refer to this booklet, then you'll be able to get a distinction. Anyway, it's M.Saidi as usual. The first thing before you go to the paper is to talk about the questions which we can't leave in the paper without asking. For example, that is the investigative question. You cannot go to the paper without knowing what is an investigative question. In this, I mean that independent variable, dependent variable, you have to know the control variables, you have to know how to state the hypothesis, you have to know how to identify the factors which make the experiment more reliable, and then the factors which make the experiment more valid. Those things in an experimental questions, you have to find them there. If the question comes, you have to find them there. So you cannot go to the paper without knowing that. That's the first point. And I have been telling you that how do I identify the independent variable? You just write ID. ID. In this ID, it means that you'll be able, I means independent variable, D means dependent variable. So it means that in most cases, the first column is dependent variable, independent variable. The other 2 or 1 is dependent variables. That's number 2, what I'm talking about. Then you also need to know that when you go for this experiment, you cannot go to the paper without knowing what we call the bar graph, line graph, and a pie chart. We will bring one. How do I identify that this is a bar graph? A bar graph, one variable must be words, for example, countries, South Africa, Zimbabwe, Mozambique, whatever. And then the next, the dependent variable must be figures. So if it is figure and figure, the first column is figure, the second column is figure, for example, 10, 5, 20, 30, 3, like that. And then the next column is 20, 30, 40. Figure to figure, that's a line graph. But if it is words to figure, then it becomes the, how do we call it? It can be either a pie chart, or it can be the bar graph. We rarely bring a histogram, but if it comes also, it is in the same case. So that is the dependent variable and also the independent variable. So how do I know? Sometimes when we bring a pie chart, listen carefully. When we bring a pie chart, yes? Please let me know in the questions. If you have any question you don't understand better, let me know very well so that I can smash it. Yes. Sometimes when we bring let me do like this so that you see me clearly. When we bring a question concerning about a pie chart, listen carefully. Don't plot or don't draw a pie chart without converting it into degrees, because a circle means 360. So if they bring things in degrees, in percentages, you have to convert it. If they bring it in grams, you have to convert it into degrees. So how do I get the degrees? You get the total, then independent, each column. For example, it is South Africa has 20%, Mozambique has 20%, and then Zimbabwe has 10%. So it's gonna be 20 out of 100 because it is percentage times 360, and then you convert it into degrees. You understand? So you cannot go to the paper without knowing that. I think I'm very clear on that. All right, let's go to now the paper, the real paper, what we expect to find in the real paper. So that is the first question. You want it or not, you will find it. Either a pie chart or a bar graph or a histogram or a line graph. You have to put the units, the dependent variable, independent variable. Those two, they must be having units. Units, you always find them in the column. You will always find them there in the column. So please make sure that you include that there. And then the title must not have any unit, but it must have what we call it must have the independent and the dependent variables there. The two variables must be shown. Don't just say a bar graph showing South Africa. No. It must have a bar graph showing maybe growth among the people of South Africa, whatever. But it must have the two variables. If one variable is not there, the answer is going to be 0. I think I'm clear there. Yes. Now let's go to what is expected in this. All right. So if you look at this, yeah, where do you find DNA? You know where you find DNA. That is the nucleus. If they ask you other organelles, you can talk about the mitochondria and then the chloroplast. Only plants, you only talk about the chloroplast, mitochondria, and nucleus. And then if they talk about animals, then you exclude the chloroplast. Then structure of DNA, the way how you see it. People are confusing what is called a nucleotide. A nucleotide is a combination of phosphate, sugar, and nitrogenous base. Please don't forget that. So we are now rushing to go through so that you don't spend more time when you are revising this. Is a phosphate, a sugar, and a nitrogenous base. Then when we go to the types of DNA, nucleic acids, these ones are easy. We don't usually ask you that, but we sometimes ask you where they are located. As I explained, the nuclear DNA, the mitochondrial DNA, and the chloroplastic DNA. Then discover of DNA. When you reach, don't waste too much time here. Who discovered the structure of DNA using X-ray diffraction pictures? Then now you talk about the Morris Wilkin and then Rosalind Franklin. Who discovered the structure of DNA? If the question is just open like that, what does it mean? If just the question open like that, you don't waste the time there. You just tell us who discovered the structure of DNA. Now you compare it who discovered the structure of DNA. That DNA is double helix with complementary base pairs. So that is James Watson and Francis Crick. Simple. Then when you go another question which they can ask you about here, yes, the structure, the natural shape of DNA. The natural shape of DNA is double helix. Don't use any other word. Double helix. Simple. We are done with that. Then you go to the structure of DNA. I told you that if you want to describe the structure of DNA, here you just look at DNA structure. Here it has it has the phosphate group, a nitrogen, a deoxyribose sugar, and then the nitrogenous bases. Nitrogenous bases are linked together with the weak hydrogen bonds, and then the nitrogenous bases include adenine, thymine, guanine, and then cytosine. Adenine is complementary to thymine. So you just, the way you see the structure, there is the way you explain it. So you don't need even to cram and then follow what exactly what I wrote here. As long as you talk about all those things which you see in different colors, then you'll be able to get the what? You'll be able to get the marks. I think I'm clear. Ne? Yeah. Let's go to another one. Then when you go, never go to the question without knowing what called base calculation. Never ever go to the paper without knowing what you call the basic calculation, because there is no way you're gonna be there and they don't ask you. There is no way. Someone is asking me that, please sir, can you do a lesson on that? I need to know what exactly you are saying there, because I don't. Please specify for me, DJ, ne? Please specify for me, and I know where exactly you are telling me to do a lesson on that. I only, oh my God, I'm seeing the time is finishing. The electricity is going. All right, you cannot go to the paper without knowing base calculation. Adenine is what? Here is showing you that adenine is, they're saying that if adenine is 30, if adenine is 20, if adenine is 10, what is the rest of, calculate the other bases. For example, how many are they? Here is simple. Adenine is 10, obvious. Thymine is going to be what? Is going to be, adenine, if adenine is 10, thymine is also going to be 10. If adenine is, if they say here on the question that adenine is 30, it means that adenine goes with thymine. So adenine is 30, thymine is also 30. So the balance, it will be for guanine and cytosine. But these two are equal. So it means that 30 plus 30 is going to give me 60. 60 minus, you subtract 60 from 100. So the balance is going to be 40. So 40 is divided by 2 so that I can get for each individual. You see? Yeah, so you cannot go to the paper without knowing this. There is something which, hypothesis and the graphs. Okay, I'll do a lesson, full lesson on that, hypothesis and the graphs. All right, thank you. Ne? I'll note it down so that you guys, I give you that lesson concerning about that. Now, when they ask you, when these calculations are here, there is something which I forgot, which we usually ask. These examiners, they like it. They like it so much. I'll talk about it. All right. So it means that if, now here is this question. If DNA has 500 bases, only 5,000 bases, only 500 is guanine. What is the ratio of thymine to cytosine? You see here, they didn't talk about cytosine, and but you know that guanine goes with cytosine. If this is 500, automatically cytosine is also 500. So 500 plus 500, 500 for guanine, 500 for cytosine, it gives me 1,000. So 1,000 minus this 5,000, so I'll be remaining with the thymine and what? I don't see the answer. Okay, I'll be remaining with thymine and adenine. So now, when I divide by 2, I'll be getting 2,000 for each, because 1,000 you subtract it from 5,000, I remain with 4,000. 4,000 is for thymine and adenine, so divide by 2 so that I get for each individual. So which makes it to be 2,000. So that is it. You see? So it means that now I can get the ratio. Please don't. Can you please do base calculation? All right, I'll do a specific lesson on base calculations. I'm just rushing. I wanted to give you this before, but the electricity is really not on my side. All right. As I'm winding up with the calculations, percentage decrease, percentage increase. Because here, I think I'll be done with the calculations within the graphs and stuff. Guys, if you have any challenge, let me know in the comment. I will do those lessons you are telling me. So I'll do a lesson on hypothesis and the graphs. I'll also do the more lessons on the basic calculations based on Kiabetri. Yes. All right. So percentage increase, percentage decrease. When this percentage increase, the formula is the same. Initial, you have to be with the initial. You have to identify where is the initial. You have to identify where is the final. I repeat, you have to identify which one is the initial, which one is the final. And then you don't stop there. Now you have to know the expression for that, of which this expression is equal to final minus initial over initial times 100. That is percentage. Increase or decrease. Final minus initial over initial. Final minus initial over you can't go to the paper without knowing those percentage increase, those percentage decrease. When the answer is positive, then it is percentage increase. When the answer is negative, then it is percentage decrease. You understand? So don't see the negative answer and then you start being worried. How comes that I get a negative answer? It means that it decreased. Percentage increase is equal to initial, is equal to final minus initial divided by initial, because this percentage times 100%. So you will be able to get the percentage increase or percentage decrease. Note on this. Don't forget that this percentage increase or decrease can be more than 100. It can be more than 100. So you can get 200% increase or 200% decrease. If it is negative, it is decrease. If it is positive, it is increase. I think I'm clear there. If you don't understand it better, let me know. Ne? Yebo. Guys, this life science is nice. Ne? It's nice. It's nice. As long as you understand what you are studying, these things are nice. You just have to know how to put love. The love will come from the subject. When you love the subject, the subject will love you back. You will see that life science, you even don't need to revise. Use the distinction material. This book, it will help you. All right. DNA replication. What is DNA replication? When does it occur? Why does it occur? And how does it occur? We are not going to ask you any other question concerning about that. When does it occur? Why does it occur? Ne? How does it occur? Yeah? Where does it occur? Those are the questions they ask you on DNA replication. There is no any other question. The only other question which can come is to bring this question, for example, the structure of DNA, and then ask you maybe is two structures, and then ask you label what is phosphate. So you cannot go to the paper without knowing how to label these structures. You cannot go the paper that this is the phosphate, this is the oxalose sugar, this is nitrogenous base, this is weak hydrogen bonds. Guys, sometimes when they bring this, this one is hidden. A is hidden, and then the B is also not shown, and then they ask you what is A or what is X. But they have given you a hint somewhere. Yes. Please, not always when there are two strands, it is DNA. No. Sometimes the second strand is RNA. If it is having uracil. Yes. So please don't confuse that. I say that on DNA replication. What is DNA replication? Yes. When does it occur? Yes. Where does it occur? Yes. And why does it occur? Those are the questions they ask you. Then, how does it occur is what you call DNA replication. Here, I'm not teaching now. Here, I'm just trying to show you DNA double helix unwind, weak hydrogen bonds break is the same thing as unzip to form two separate strands. One strand or each strand is acting as a template using free-floating nucleotides of DNA. Yeah, from nucleoplasm, a complementary strand is formed whereby adenine pairs with thymine, guanine pairs with what? With cytosine. Two new DNA are formed which are genetically identical. Guys, if you have any other question which you want me to repeat in my free time, let me know. Ne? Yeah. And if you want more lessons which are live like this, where you can even ask you questions, let me know in the comments below. Ne? Yeah, let me know so that I can organize for you guys these classes. Ne? Yeah. DNA profiling. This one is simple, very simple, but in most cases, people, they tend not to do it. And I think I've never seen any question paper without DNA profiling. I've never seen it. Yes, so make sure that at least you know when you go to the paper, when you know the uses of DNA profiling, and also how to tally to know which one is which one is a criminal, yes, or a biological father or what. Yes, disadvantages, I think that one is also clear. Types of RNA. We don't usually ask you types of RNA. We only ask you types of RNA when it comes to protein synthesis, and then we ask you that, for example, which one we X, Y. For example, W is DNA, and then Y is the amino acid. X is messenger RNA. You see, that's a type of RNA. And then also, if you look at here, this one which looks like a T is a tRNA. Then transcription, translation. So if they don't bring the description of transcription in full, then we can bring in this format. Yes, so we can bring this in this format. So it means that that's how you can do that. So DNA transcription is simple. Transcription, the way you see it here, I'm not going to keep on repeating, but different colors means a marking point. Guys, let me know if you understand what I'm saying. Yes.